This is the first part of notes on Unit 11, which is viruses and bacteria. Viruses are covered in part of Chapter 10 in your textbook, so if you need to go back through and read some of that, that's where those pages are located. So this is a picture of a herpes virus. A herpes virus is a class of viruses that includes things like the virus for chickenpox, the virus that causes fever, blisters, and cold sores. And they aren't really this color. The color has been added into the electron micrograph to illustrate different parts of the virus. Viruses are what we call acellular, which means they're not made of cells. They are parasites of cells because they can invade and reproduce in cells, but they're not cellular, and therefore, by the definition that we use for living things, they're not really a living thing. They're not classified in a domain or kingdom because they don't fit the characteristics of the living things that belong in domains and kingdoms. They're very small, on the order of 17 to 1,000 nanometers in size. They don't really do anything. They don't metabolize nutrients. They don't use or produce energy. All they do is invade cells, take over the cells to make them reproduce the viruses, and then they kill the cell. We usually classify viruses by one of three ways, or a combination of these three, rather. By genome, which means what their nucleic acid is. They either are DNA or RNA viruses. The RNA viruses are called retroviruses. We'll talk about why that is in a few minutes. But they're also classified by what the virus might infect, whether it's a plant virus or a bacteriophage, like it says here, that infects bacteria, or an animal virus. Some viruses are specific to certain species or certain types of organisms, and others can go between different species. The third category that we use to classify viruses is the shape, and you see here the three main shapes, which are spherical, polyhedral, and helical. And then you have some more complex ones, like we see the bacteriophage on the far right, that is a combination of two or more of those shapes. This one is a combination of with a polyhedral head and a helical tail. And the tail fibers that you see there that look kind of like legs aren't really legs, and they're not used to walk with. They're used to help attachment to the cell that they infect. Viruses basically are genes in a box. Basically, you've got a polypeptide or protein coat called a capsid that contains a bit of nucleic acid, either RNA or DNA. Sometimes there is a membrane envelope around some viruses that is derived from the cell membrane of the host cell. But that's really about it. There's not a whole lot to them. Now, viruses have two different types of reproductive cycles. When you have an active virus infection, that's called a lytic cycle. In the lytic cycle, the viral particles are produced by the cell. Basically, they inject their nucleic acid, or they get their nucleic acid into the cell. That takes over the cell and uses the cell machinery, so to speak, to produce the components of the virus. Once the virus particles are manufactured by the cell, the host cell lyses or splits open, ruptures, and therefore dies, and that releases more viruses to the environment to infect other cells. The diagram here shows you the basic stages. This is a bacteriophage infection, showing how the bacteriophage injects its DNA into the cell. The DNA basically takes over the cell, causes the cell to produce the proteins and the nucleic acids and the other parts that go together to make the virus, and assemble them into finished viral particles. Once that's finished, then the cell lyses, and that releases the phages to attack other cells. The other type of viral life cycle, or not life cycle, but reproductive cycle, is called the lysogenic cycle. In the lysogenic cycle, the viral DNA is inserted into the host chromosome, not just into the cell, but into the chromosome. The process of inserting DNA from a different source into a chromosome is called recombination, and it kind of hides there. It's kind of a stealth mode, I guess, is the best way to say it. Every time that cell reproduces, then the viral DNA is also duplicated along with the host chromosome. We call that inserted DNA in a bacteriophage, a prophage. in a eukaryotic cell, we call it a provirus. Most of the genes in the prophase are inactive, but the virus may become active at a later date due to some kind of environmental signal that can vary between a lot of different things. But basically, whatever the signals are that can stimulate this can cause a switch in the reproductive cycle to the lytic cycle, and that will cause the viral DNA to separate from the chromosome, become active again, and enter into the lytic phase. And just remember that since every cell that has been infected the virus is reproduced every time that cell undergoes mitosis, then you may have a lot of cells becoming active all at the same time. Here's the lysogenic cycle and a bacteriophage. So we've got our phage DNA inside the nucleus like we had before, but if it goes into the lysogenic cycle, then basically the DNA gets inserted into the chromosome of the bacterium, and every time the bacterium undergoes cell division, every daughter cell that results also has the virus DNA in it. Some kind of environmental stress may cause the DNA to leave the chromosome, and then it'll enter into the lytic phase again. And many viruses do this kind of thing. They can't all do that. Some are only lytic viruses, but others are lysogenic. For instance, the fever, blister, cold sore, herpesvirus becomes inactive in that lysogenic phase until stimulated by some environmental stressor that causes it to become active again, and that's when you break out with a fever, blister, or cold sore if you are infected with that. Viruses that appear suddenly in populations or are new to physicians or medical scientists, these are called emerging viruses, and ones that we've heard a lot of in the last 30 or 40 years are the AIDS virus, which is called HIV, the Ebola virus, West Nile, Zika virus, and SARS virus. So lots of different things that come from various sources. Usually they're either the result of a mutation that makes them become infective, or the result of contact between species. Some of these viruses were first found in animals and then later became mutated so that they could infect humans as well. And the other reason that we often see emerging viruses is because they end up being spread by from isolated communities or isolated populations to larger populations. That's what happened with the Ebola virus a few years ago. Ebola's been around for 40 or 50 years or so. Medical scientists have known about it and known what it did for that period of time, but it wasn't there wasn't a big outbreak of it until just several years ago, because before that it was found mostly in isolated villages, and then it would just it would run its course and kill a lot of people, but some people always survived, and then it would basically burn out and it'd disappear for a period of time until it came back again, probably in another village. A couple of years ago, by the time we had the big outbreak a few years ago, it was because a lot of people had moved into cities and would go back to their villages to visit and bring the virus back to the city and spread it among people. And then somebody could get on an airplane and fly to some other part of the world, and we ended up with a much bigger outbreak than had been seen in past. Here's another virus that's well known in the past few years. This is the H1N1 virus that caused the really bad flu in 2009 that a lot of people were affected by. It was a pretty bad flu, and it was one that was not included in the flu vaccine that year because they hadn't anticipated the change in the virus that came at that point. AIDS is one of these retroviruses. Okay, AIDS stands for acquired immunodeficiency syndrome, and it's caused by a virus called HIV, human immunodeficiency virus. This is an RNA virus, which is also called a retrovirus. It has 2 copies of the RNA genome, and it also has 2 copies of an enzyme called reverse transcriptase. What reverse transcriptase does is it makes DNA from an RNA template. This is why RNA viruses are called retroviruses, because they work backwards, because retro, contrary to what you might think from the way it's used, retro doesn't mean old, it means reverse or go backward. And so the RNA gets copied into DNA or reverse transcribed into DNA, and then a complementary DNA strand is made, and then that can take over the cell. Here's kind of what, here's a diagram showing what the HIV looks like. There is an envelope with glycoproteins on it, and the envelope and glycoproteins are derived from the host cell. Inside the envelope, you've got the protein coat or the capsid surrounding the genome. We have 2 identical strands of RNA, and we've got 2 copies of reverse transcriptase enzyme. And so it's basically a package ready to be delivered to cells to create havoc for cells. The way it works, the reverse transcriptase makes a DNA strand from the RNA, and then it adds a complementary DNA strand. It can then integrate into the chromosome, becoming a provirus, like we said before, and then that can produce messenger RNA to translate the messenger RNA into the proteins that make up the virus, and then assemble the new viral particles and leave the host cell. Sometimes it doesn't kill the host cell, but it can then infect other cells. And when you have a lot of cells that are infected, then that will definitely cause problems, because the HIV virus attacks the immune system cells, and that causes the problems that result from AIDS. AIDS is not the disease that causes you to die from it. It is the secondary infections, or the infections that you would not get if your immune system was working properly because of the virus. Here's the infective process that we can see here. There's the viral RNA with the reverse transcriptase enzyme, which makes the DNA strand, and then synthesizes the double-stranded DNA that injects into the chromosome, and then it can produce other cells. And you can see where the viral envelope comes from the cell membrane itself. There are some other things that are also not living that can cause various kinds of diseases. One is called a viroid. Viroids are small circular RNA molecules that infect plants, and they can interfere with plant growth by replicating inside the host cells without producing proteins. The other kind of infectious protein is called a prion. Prions can cause degenerative brain diseases in animals. This is the source of the disease you may have heard of called mad cow disease, which doesn't make the cows mad or crazy, but they do start having some degenerative muscular and neurological symptoms. Apparently they are misfolded forms of normal brain proteins, which can then convert the normal protein to a misfolded form.